right, chapter 3, biological macromolecules. So biomolecules, biological macromolecules, organic macromolecules, there's a lot of terms that mean the same thing. These are molecules, all organic molecules contain carbon. So the word organic in this context refers to carbon serving as the backbone of the molecule. Typically you have hydrogen, oxygen, nitrogen, and then some other things, maybe the occasional phosphorus or sulfur, for example. The carbon's going to be the backbone. More than one carbon linked together creates the backbone of the organic molecules. So in this chapter, we're going to dig down into the 4 classes of biomolecules. And I'm not going to lie to you, this chapter has a lot of material. So be ready to spend some time on this and really commit this to memory. You're going to walk into your next course or courses in the future, and they're not going to go through all this again. They're going to expect that you know the fundamentals, and these are literally just the bare basics of carbohydrates, lipids, proteins, and nucleic acids. So it's going to feel like a lot of material, but it's really just the basics. So what are macromolecules? They're huge molecules composed of thousands of atoms. In cells, macromolecules are typically monomer units that are joined together one after another to make what we call a polymer. So really, for most of the macromolecule groups, once you learn the structure of the monomer units, the polymer is just that same monomer unit, just repeated over and over again, linked together covalently. Ultimately, the shape of the final structure dictates the function. So structure and function are very key concepts in biology. So the way that any polymer is formed is by a reaction referred to as a dehydration synthesis reaction. So dehydration synthesis reactions join monomers together, and they release, or as part of that joining process, there is some water that is produced as a byproduct. So you'll see in this picture at the top of this slide, this shows 2 monomers, and you see the little plus sign in the middle. So this one on the left and this one on the right, and then the arrow, that means bonds are forming and bonds are breaking, because remember our chemical reaction has that arrow that shows that. And then on the right side of the screen you see the product. In this case, everything is joined together. Well, the 2 hexagon-looking structures are joined together, and then a water is formed as a byproduct. So this is the general way that all monomers are joined together to make polymers. It's called dehydration synthesis. There's always a water that's formed and is released from the reaction. Sometimes this reaction is also called condensation reaction. So dehydration synthesis or condensation reactions are the same kind of same type of reaction. Those are synonyms. There will be enzymes, we'll talk about what enzymes are, that help to facilitate the making and breaking of bonds. And so that's an example of dehydration synthesis or condensation reactions. So these build polymers. Now, the opposite reaction is called hydrolysis. That's the process where water and a polymer get recombined, and it causes the breakage of bonds that hold the monomers together. So basically you take this reaction that you see on the screen, and it's exactly the same, just turn the arrow the other way and you will have a hydrolysis reaction. So sometimes monomers are joined together to make polymers. Other times polymers are broken down by the addition of water to make monomers. So both things will happen in the cell. The cell is constantly building and breaking down different polymers from the monomers. All right, so water is needed to as part of the hydrolysis reaction. All right. So some prefixes you have to know. Mono is 1, di is 2, tri is 3. Oligo, you'll see that occasionally, means a few. And poly means many. All right, so just some terminology I want to make sure you know. So we're going to go through the first, actually I like to teach the carbohydrates first, and then I like to teach the nucleic acids. So we'll do those two groups first. And then we'll take a break in terms of the lecture. And then in the second part of the lecture, we'll go over the lipids and the proteins. So first thing to do is to memorize the 4 types: carbohydrates, nucleic acids, proteins, and lipids. Make sure you know the names of the 4 main groups of biomolecules. So now we're zooming in, looking at the carbohydrates specifically. Carbohydrates are a group of molecules that really have a fairly common, a very predictable structure. They always have a molecule structure that is some multiple of 1 carbon to 2 hydrogens to 1 oxygen. And if you look at that, it's 1 carbon and then a water in a sense. So that's where you get the name carbo, that refers to carbon, hydrate, that's water, hydration. It's the hydration of carbon or something. But anyway, so most of your sugars will have some kind of multiple of this basic molecular structure. All right, so monosaccharides, those are the monomers for this particular category. Each category, carbohydrate, proteins, and nucleic acids, will have monomers. Lipids don't have monomers, but the other groups do. And in this case, the monomers for this carbohydrate category are called monosaccharides. Mono means 1, saccharide means sugars. So these are single sugars. And then polysaccharides would be if you have lots of monosaccharides that get joined together by those dehydration synthesis reactions, then the resulting molecule is called a polysaccharide. All right, so the simple sugars, the monosaccharides, are the monomers. They're the building blocks that get put together by those dehydration synthesis reactions to make the polysaccharides. And all of these are part of this carbohydrate category. So monosaccharides, like we said, have some kind of molecular formula that's some kind of multiple of this basic CH2O. So for example, the picture that's shown here, the glyceraldehyde, which is on the left, that one has C3H6O3. So you can see it's just a multiple of the basic formula. The names of monosaccharides usually end in the letters OSE. So you have glucose, but in some cases you don't. So what's kind of funny is the examples that are on this page don't necessarily follow that rule. But most of the ones that we're going to look at, all the ones that we're going to look at that are monosaccharides, the ones we're going to look at in a significant way are going to end in OSE. So that's a hint. Anything that does end in OSE is a simple sugar. And by far the most important monosaccharide that you're going to get to know very well is glucose. And the formula for glucose is C6H12O6. So glucose has 6 carbons, and it's shown here on the left. It has a backbone of 6 carbons, all right? You can count the 12 oxygens, you can count the 12 hydrogens, and 6 oxygens. There's also a simple hexose sugar, hexose means 6 carbons, called galactose, and one called fructose. And you remember isomers are slightly different versions of molecules that have the same formula. So all 3 of these have the same formula, C6H12O6. You don't need to memorize the structure for galactose and the structure for fructose, but you will memorize the structure for glucose. So that one I will require. All right, but we're not going to memorize the linear structure, which is what is shown here. We're going to memorize what we call the ring shape, the ring structure of glucose. So what happens with glucose is, and what happens with all the sugars, is they can make another bond and kind of close up. Instead of being a linear shape, they can close up and form this ring shape. So the ring shape is, for us, going to be the more significant shape that we're going to be seeing when we look at the glucose. So the ring shape of glucose goes like this, and I need a pen, so let me get a pen. All right, so you see the oxygen, there's an oxygen here, and this is kind of the standard way we write it. And then when you have when you're drawing structures in organic chemistry, anytime you have like an intersection point or a corner, if there's nothing marked there, then that's a carbon. So where I'm putting, now this is an oxygen, they put an O there, so that's not a carbon. So right here is oxygen, but where I put the closed dots, those are, you are supposed to know that when there's just a corner or an intersection of lines that doesn't have a letter, that's assumed to be a carbon, because carbon is very common, of course, in organic chemistry. So instead of drawing out all the carbons, they just use this shorthand, but I'm going to draw them all in. So you have an oxygen here, and then as we go around the ring in a clockwise way, and you're learning some organic chemistry here that's going to help you, we number the carbons 1, 2, 3, 4, 5, and then we have one carbon, and really this bond goes to the carbon, not to the hydrogen, but it's, you know, they're kind of assuming you know that, but this carbon here, they actually write the carbon on that one. That's the 6th carbon. So the 6th carbon is not considered part of the ring. It's kind of funny because on this drawing they did go to the carbon, so that looks better. Same thing here, 1, 2, 3, 4, 5, and 6. Okay, so that's how you count it. Same thing with like fructose and ribose, which are down here. You would count around the ring. Actually, it ends up being 1, 2, 3, 4, 5, and 6. But you don't really need to worry about these guys. So glucose, you do need to start to recognize. I'll just circle this one up here. And you do have to know how to count the carbons. So when the oxygen is sort of in the upper right-hand corner of the hexagon, then as you move around clockwise, carbon number 1, 2, 3, 4, 5, and 6 go clockwise, and then the 6th one is the one sticking out. All right, so that's the ring structure of glucose. And what you'll notice about the ring structure of glucose is that each of the carbons has a hydroxyl group attached to it. You can see the hydroxyl group. Now, the 5th carbon does not, but all the other carbons have a hydroxyl group. So that's a very important characteristic of glucose, because that those hydroxyl groups are going to be important for interacting with other glucose or other sugar molecules for that dehydration synthesis reaction. So for example, you can see here on the left you have glucose, and then they added a little fructose here. But what's going to happen is these two molecules, glucose and fructose, and then here's the arrow for the reaction. So that's the reaction. And then that's going to join together. The reaction is the dehydration synthesis, and it's going to then join together that glucose and that fructose. So instead of having one molecule at the end of the reaction, instead of having 2 molecules at the end of the reaction, you just have one. But that new molecule is called sucrose, and it's really just a glucose that's covalently joined to a fructose. But now the whole thing is just one molecule. And then what they didn't show, which they probably should, is the water molecule that's released, because this is a dehydration synthesis reaction. They're just showing it vertically in this slide, and earlier they showed it drawn horizontally, but that doesn't matter. So the bond that forms when two simple sugars are joined together, the bond that forms between them is called the glycosidic bond. Sometimes it's also called glycosidic linkage, but the word glycosidic is kind of your key. That's really the giveaway. So the glycosidic linkage is something that forms when sugars are joined together covalently. When you have a very long chain, more than just a few sugars joined together, we call it a polysaccharide. And there's a few types of polysaccharides that you want to focus on. And some polysaccharides are in animals, some are in plants, and those are different, slightly different. They're all made of glucose. Most polysaccharides that we're going to discuss are made of glucose. And that's why I want you to memorize the glucose structure specifically. And some of them form structural parts of the cell, like rigid structures that hold things, serve structural purposes in the cell. And some polysaccharides are used for energy storage is what we say. And so those are really the two things, plant or animal cells, and is the polysaccharide used for energy storage or for structure? All right. So polysaccharides that have energy storage functions, one is called glycogen. That's only in animal cells. And then plants have starch, and that's the energy storage form. So if we're talking about energy storage in animals, we would refer to glycogen. If we refer to energy storage in plants, we would refer to starch. Now, both glycogen and starch are made up of many glucose monomers that are joined together by dehydration synthesis reactions, and that creates a whole bunch of glycosidic bonds. And then those structures form these coiled shapes. So we'll see some pictures of those in a minute. Now, there are some polysaccharides, like I said, that serve a structural purpose. In plants, we call it cellulose. And in animals, specifically insects, and also in fungus, we have a structural polysaccharide called chitin. I'm going to give you guys a hint. Anything in biology, I don't know why my mouse is doing this. Anything in biology that has a CH always has a hard K sound. So you would not say chitin, you would say chitin. So anything with a CH in biology, let me think if I can think of anything that's an exception to that rule. 99 times out of 100, it's going to have a hard K sound. So being able to pronounce the words helps you to, increase your expertise in that subject. So CH is going to be a hard K sound for most of the words. I'm sure there's one exception, but 99 out of 100 times it'll be a K sound. So cellulose and chitin, they don't form coiled shapes, they just form straight fibers, like rope. Rope literally is cellulose, and it literally makes these straight, strong fibers. All right, so let's talk a little bit about each one of these. Glycogen is made up of glucose monomers. And there's no limit on a polysaccharide, there's no limit on how many monomers it is. Okay? So, it's usually hundreds, could be thousands. There's no absolute length. I guess after you get, you can say I have a monomer, I have a monosaccharide, I have a disaccharide. I guess you could say I have a trisaccharide. And then after it gets to be a certain length, you just say polysaccharide. And that covers all kinds of different lengths. All right. Most of your glycogen in your body is stored in your liver cells and in your muscle cells. And glycogen is typically very branched. In other words, we'll look at some pictures of it in a minute, but you have all these, you can see all the little glucose hexagons in the structure, but then some of the hexagons branch off in different ways. So branched. All right, here's this is starch. Starch is the energy polysaccharide, energy storage polysaccharide of plants. Some starch is branched and some is not. So this would be down here, shows branched. So you have all these monomers joined together, and then you have kind of a branch over here. And then they show it kind of more in a, and you can see that it's coiled like that. All right. So starch can be branched or unbranched. Glycogen is always branched. But both of them are made of glucose monomers, and both of them will coil in their structure. Now, what about the structural polysaccharides? You have cellulose and, like I said, chitin. Cellulose makes up the cell wall of the plant cell. And in fact, all cell walls, because plant cells have cell walls, so do fungal cells, so do bacterial cells. Really, animal cells are the ones that don't, primarily, but a lot of organisms have a cell wall, and in plants it's made of cellulose. In other organisms, it's made of something else, but it's always a carbohydrate. But in this case, we're just going to talk about the plant cell wall is made of cellulose, and cellulose is a polymer of glucose again. And it makes a linear chain. It doesn't spiral, okay? And so you end up with this long, straight, fibrous structure instead of this spiraling structure. And it's also not branched. So unbranched, that's a rule here. So it's just a straight chain. I'm going to try to write unbranched. So starch can be branched or unbranched. Glycogen is always branched, and cellulose is never branched. And then the last one is chitin. Chitin makes up the exoskeleton, the hard, crunchy part of the insects. And for the fungi, it makes up the cell wall. And fundamentally, chitin is a polysaccharide. It's not just glucose, though. You can see the glucose structure here and here, but there's some things attached onto it. So we call it a modified glucose. So it's a modified glucose that make up the monomers that are joined together to make the polymer that we call chitin. All right. So you want to, I think what's helpful when you have all that information that I just gave you is to try to create maybe a table. All right. I'm going to exit out of this. Let's see. So let's create a table. I'm going to insert a table. And I want to have monomer type. Let's do that. Okay. I need one more. Oops. Let me get rid of this. All right, insert table. Okay, that's better. Okay, so polysaccharide name. And then monomer type. So made up of what kind of monomers. And branched or unbranched, plant or animal. Plant or animal. And then I probably also need a, we can add that in later. All right, so The polysaccharide that is glycogen is storage. That's a storage polysaccharide. I'll just add it here. All right, your monomer type is glucose. Branched or unbranched is branched. Plant or animal, animal. That's the one that's only in the liver. Well, it's not only, but mainly liver and muscles. The other storage polysaccharide is starch. And that is found in plants. It's made of glucose. It can be branched or unbranched. So there's a couple different forms. So there's some types that are branched, some types that are unbranched. And then that is plants. All right. Now for your structural polysaccharides, you have cellulose. So that makes up the structure, remember, the cell wall of the plants. And that's made up of glucose. Oops, gotta get the caps on here. Glucose. And cellulose is unbranched. And that is plants. And then the other structural one is chitin. Now this one is the only one that's a little bit different. It's the modified glucose, the one that had a little extra something there. Had a little nitrogen in it. Chitin, I think chitin's unbranched. And animals and fungus. All right. So, you have all those slides and then you can kind of put it together and create a chart that helps you to study. It's very helpful because as you're going through slides, and this is true in any science class that you do, you're like, okay, how can I reorganize this information in a way that I can see it side by side? I can see that glucose, you don't need to hardly memorize that because it's all the same, but the other things are different. So those are where you want to pay attention to, what do I need to know? All right. Okay, so we're going to move on to nucleic acids. Nucleic acids store and transmit hereditary information. There's DNA and there's RNA. Nucleic acids can be found, both DNA and RNA are found in the nucleus of the eukaryotic cells. For eukaryotic cells that have mitochondria and chloroplasts, they are found there as well. Prokaryotic cells don't have a nucleus, so the DNA and the RNA are just in what we call the cytoplasm, just in the cell. All right. So the monomer units of the nucleic acids are called nucleotides. But here's what gets a little complicated. The nucleotide has parts. So the monomers are the nucleotides, but each nucleotide has 3 parts. A pentose sugar, that means a 5-carbon sugar. So you will see something that looks a little bit like the glucose that we looked at earlier, but it's instead of being a hexagon, it'll be a pentagon. And you can kind of see that over here. Let me get my pen. You can see the pentose sugar over here. And then a nitrogenous base, we'll talk a little bit about what that is. And then a phosphate group. Now hopefully you know now what a phosphate group is if you memorized your functional groups. So, all right. So a 5-carbon sugar, we're going to practice recognizing sugars. And a phosphate group, hopefully you can recognize that. So the only thing that's going to give us a little bit of difficulty is the nitrogenous base. But notice what's in here, nitrogenous base. A nitrogenous base contains nitrogen. So that's going to be a good clue for us. The nitrogenous bases are shown here, and you've probably studied a little bit about DNA and RNA before, so you might recognize the nitrogenous bases have ring structures, but they're not glucose. So once you memorize the structure of glucose, you'll be able to look at these guys and say, that doesn't look the same. I mean, it's a ring structure for sure. These ones have 2 rings fused together, but there's a bunch of nitrogen in the ring. So you guys see the nitrogen in the ring? So that's these are nitrogenous bases. I'm not going to ask you to know which ones are which, but these are nitrogenous bases. Okay, see all the nitrogen in there? So look for that. So when you have a 5-carbon sugar, a phosphate group, and a nitrogenous base, that makes up one nucleotide. And then if you hook together, you know, hundreds of nucleotides into a straight chain, it doesn't branch at all, then you end up with a polynucleotide, or we just call it a nucleic acid. All right. So for right now, we're not going to dig down into some of these details, but in later chapters, we will look more closely at the structure of the nucleotides. So for right now, we're going to look at the general structure, very generally. All right. Now, RNA, not always, but often has a single strand, a single polynucleotide strand. DNA, often, but not always, but usually has 2 polynucleotide strands that connect by hydrogen bonds. Remember hydrogen bonds, those weak bonds that occur between the partial positive end of one molecule and the partial negative end of the other molecule. So the hydrogen bonds are in the center, and then the whole thing twists. That concept of spiraling is very common in the different macromolecule groups. Okay, so we're going to do a little practice on identifying the molecules. And I put up a little practice packet for you, which I am now going to go to. And I want you to look at the practice packet. And the first page of the practice packet says Carbohydrate nucleic acids, part 1. Hang on. I just want to go back. No, I don't want to do that. All right. How do I zoom out? Oh, here we go. Okay, here we go. Okay, so let's say we're looking at this. All right. The way I would do this if we were in a lecture class, I would say, okay, look at the screen here. Look at the worksheet. And what I want you to do first is find glucose. Find glucose. Just one monomer unit of glucose. And students, you look at each one of these, and most students will be able to find this, but you take your time. And this first number 1 is glucose. All right. So look at that. Look at the glucose. It's a hexagon with 1 carbon kind of sticking up out. But usually when we get lazy, sometimes we just draw glucose as a hexagon. Glucose is so common in biology that if they just draw a hexagon, you can assume it's glucose. So if it's not glucose, they need to tell you, because hexagon means glucose in biology and organic chemistry. So number 1 is glucose. Okay. Now, what else do you have on the screen that has hexagons that are glucose? So number 3, if you can see it, it's a lot of hexagons in a chain, and they're trying to show you that the chain is spiraling. So what could that be? It doesn't look like it's branched. Sometimes there's more than one right answer. That could definitely be starch, because starch spirals and it's not branched. Okay. If it was branched, it could still be starch, or it could be glycogen, because glycogen branches. And remember, the chitin and the cellulose don't spiral. They're straight. So I would go with starch on number 3. You don't have to get more specific than that, just starch. If you just put polysaccharide, it depends. If it's a multiple choice question, pick whatever's appropriate. If it was a free answer, I would accept starch, I would accept polysaccharide, Okay, number 4, what is number 4? Well, you do see a glucose here, but then it's connected to another simple sugar, so that's a disaccharide. 2 sugar units together. So you could just put disaccharide. And then the glycosidic linkage is this little oxygen in the middle between the 2. that's holding them together. That's the glycosidic linkage. All right, you have another sugar down here, but this is only a 5-carbon sugar, but it's still a simple sugar down here on number 6. Then you have number 5. What do you think number 5 is? It looks like little hexagons joined together, but there's kind of a little branch. It kind of makes a little fork in the road here. It's not very long, but you could put polysaccharide. You could say, since it's branched, it could be glycogen, it could be a piece of starch. I mean, it's just a little piece. So there's a lot of correct answers that you could, that would fit. So, first look for the simple sugars and the polymers of the sugars. So number 1 is just plain old glucose, monomer. Number 3 is a polymer of glucose, and since it's spiraling, we're going to go with starch. Number 4 is 2 sugars joined together. That's a disaccharide. Number 5 is several sugars joined together. You could say polysaccharide. You could say starch. You could say it could be glycogen. You can't tell for sure. Something like that. On number 6, that's a monosaccharide. Even if you don't know the name, monosaccharide, simple sugar, something like that. And then that leaves number 2. What is number 2? It has ring structures, but it doesn't look like glucose. So it's the thing we just went over. It's the nitrogenous base. So this one has 2 ring structures fused. Some of them just have 1 ring structure, but you see a bunch of nitrogen. Nitrogenous base. So that's what I would put there, nitrogenous base. So I'm asking you to recognize, I'm giving you the picture and you're telling me just in general what it is. That's a nitrogenous base. It's not glucose. It's not a polysaccharide. Okay. So the more we practice these, the better. And you have the packet, so you have a blank copy. You can keep practicing. Okay, number 5. Oh, we did that one. Number 7. Number 7 has a phosphate group. It has a sugar. And it has, this is a different nitrogenous base, but it's another nitrogenous base. It has all 3 components of a nucleotide. So it's a nucleotide, which is just a single unit. And number 8, I don't know if you can tell, but there's 4 nucleotides joined together in a chain. So you could call this a polynucleotide chain. You could call it a nucleic acid. I think that's what you could call it. All right. So I'm asking you to be able to look at something and, like, if I showed you number 8 on the test and I said, is this A, glucose? Is this B, and I'm talking about the whole thing, is this B a phosphate group? Is this C, polynucleotide chain, polynucleotide? Is this D starch? So you want to be able to narrow it down to, okay, this is several nucleotides joined together in a chain. That's a polynucleotide or a nucleic acid. That's what I'm asking you to do. So there's another page here. Let's see how we can do on this page. So again, similar. All right. All right, I'm going to give you the answers, but what I think would be helpful is that you try to do it on your own before you listen to the answers. Because you won't know if you know it until you can do it without me telling you the answers. Okay, so you might want to pause right now and see if you can do this one. Write it down on a piece of paper and then unpause me and see if you got the right answers. So number 1 is glucose. You should be able to recognize glucose. Number 2 is ribose, or if you don't know the name of the sugar, you can just put monosaccharide. Glucose, I expect you to know the name of, but number 2, you can just put monosaccharide. Number 3 is 1 nucleotide. It has all the 3 components of a nucleotide: phosphate group, 5-carbon sugar and nitrogenous base. So altogether it makes a nucleotide. Number 4 is a nucleic acid or a polynucleotide. Number 5 is a polynucleotide. You may or may not be able to tell one is DNA and one is RNA, but at this point I'm not that concerned with being able to tell that difference. So 4 and 5 are both polynucleotides or nucleic acids. Number 6 is a nitrogenous base. Number 7 is a disaccharide, 2 glucoses joined together with a glycosidic bond. And number 8, I don't know if you can tell, but those little red things are hexagons. So it's a highly branched, spiraling hexagon structure. So it could be starch, it could be glycogen, or you could just put polysaccharide. That would work. Okay, so that is where we're going to stop with the practice packet right now. In the next lecture segment, we will go over the other types of macromolecules, and then we'll do some practice packet sheets that have all of them on there. And the more you practice these, the more you're going to be surprised that you're going to be able to start to recognize one from the other, even though right now you think I'm crazy. We're gonna practice what are you looking for so you can tell which structure is what. All right, and we're just trying to very generally identify them right now. All right, this is good fundamentals for you for your organic chemistry class, which I know you think is a long way off, but it's going to come and creep up on you. All right.